All right, trying to get some information for you here, guys. We left off in notes, kind of right around here, I think. And I want to pick up where we left off and just give you a couple of minutes here of what you missed. We talked about the 9 plus 2, and we talked about how those cilia and flagella move. And we also talked about how sometimes those microtubules are used for cell division. Next thing to talk about then, moving forward, would be microfilaments. So they're the next group. So that would be the next thing that you'd want to take notes on. Here's a quick version of it. You can pause as you need to. So microfilaments, really, really small things. They're made out of actin, which is a protein. Here is a quick description of them. Those actin filaments are basically like, I don't know, you'd think of them as like long dental floss types of materials. So pretty thin and weak all by themselves, but collectively they can do quite a bit of work. They're involved in the shortening and lengthening of cells. That's what we know about them. And muscle cells, which need to contract and lengthen, and amoeba are used extensively for these things. So here's what an amoeba looks like as it moves through a system. So here's that little amoeba cruising along, and it ends up, this is at sped up pace here, but an amoeba is like a little blob, a single-celled organism, and it moves by actually oozing these little microfilaments and pushing them against the cell membrane from the inside. And it basically swells out one part of its body at a time. So that is what you're seeing here, is this little faded kind of shady, hazy area here. These are all just microfilaments just being pushed in this direction, squeezing out the plasma membrane so it can move in that direction. So that's how that works, those little filaments. The other thing we know about microfilaments is that they make up something called the matrix. And the matrix is located just inside the plasma membrane, so just inside the cell membrane. And we think it has something to do with water regulation. It's that little thin layer on the inside of an eggshell. An egg is actually a single cell. And this is basically the same exact thing we think on all cells out there in nature. That there's this little microfilament layer that's just meshed together that basically helps control water regulation, we think. All right. And then the final group is intermediate filaments. These are all kinds of different degrees of diameter. Basically, we've clumped the whole rest of everything else into this group. One thing that they usually use as a standby is that they say that they have to be made out of keratin. And keratin is what makes up your like fingernails and your hair. It's a protein found in there. Really, really durable. So like you know that after someone passes away, that their hair and their fingernails usually remains for a very, very, very long time. It can be actually like thousands of years even. So that keratin, nothing out there in nature has an enzyme to break that down. So really, really strong, durable, lasting material. So these structures are considered to be pretty permanent. So again, they're made out of keratin, these intermediate things. And basically when a cell, like you leave a cell behind on a desk or on your bed or on a table or on a chair or whatever, all the components of it, the fats, the proteins, the everything, that all breaks down, but the keratin remains. And that's what makes up dust. Okay. And then recently, about 5 years ago, University of Minnesota was doing some research on this, and they came up with some ingenious ideas. And what they did was they said, well, the best case scenario for like organ donation would be if you could get your own organs. But you can't do that, obviously. But what we can do is we can take some other organism's organ, and we can wash it of all of its cellular material. And you literally use like detergent, like Tide laundry soap, modified a little bit, honestly. And what they end up doing is they rinse off, like this is a pig heart, and they rinse off all the cellular material by just washing it with detergent. The only structure strong enough to remain are these intermediate filaments here. And what they end up doing, this is just a keratin shell of the heart then. So there's really nothing, there's no cellular material here at all. Then what they end up doing is they take human stem cells from the donor himself or herself, and they spread them onto these organs, and those stem cells recognize, oh, I'm part of a heart. And so they start developing into heart tissue, cardiac muscle, connective tissue, etc. and they actually start to reestablish this whole thing as a viable organ that can be reanimated at that point. Really, really fascinating stuff. And then that guarantees that you don't get an organ rejected by a donor because it's that person's own cells. So really, really fascinating stuff. But thought I'd just give you an idea of how keratin and how these intermediate fibers or filaments are being explored right now. So those are the three then. We have our microtubules and our intermediate filaments and our microfilaments. Those are the three that you need to know about. That's all from chapter 6.6, and I think that's all you need to know for this point. All right, thanks.